This is a drawing of the cell membrane. You can see two parts to the membrane, the phospholipid bilayer and the proteins. The phospholipid bilayer has two parts. The pink circles are the heads of the phospholipid molecules. The gray tails are the tails of the phospholipid bilayer. Notice that the heads are at the top and the bottom of the membrane, and the tails face inward. The blue molecules represent proteins, and you can see three basic kinds of proteins. The protein on the left, that's only on the top of the membrane and has that chain of red shapes coming out of it, is a marker protein. Marker proteins, remember, identify the kind of cell. On your drawing, you'll see marker proteins as proteins that do not pass all the way through the membrane, but only come out and stick out the top side of the membrane. The middle blue structure, the one that's a solid oval, is a receptor protein. It passes all the way through, but it looks like it's a solid molecule. There's no gap in it, no pore or channel in it. And this is the kind of protein that attaches to signal molecules. And then finally you see the blue protein that has the white stripe down the middle. That white stripe represents a channel. And this is a transport protein. It has a kind of pore that passes all the way through the membrane so that particles can pass from outside to inside the membrane or from inside to outside the membrane. Now I'd like you to look at the handout that you've been given. On page 1, there's a drawing of the cell membrane. There are lines pointing to the 6 structures that I just mentioned. I would like you to label these 6 structures. You can see the names of the structures in the bottom left-hand corner of the page. Print the names of these structures next to the line that points to the drawing of those structures. I think you should be able to recognize the cell membrane, that's the whole membrane, the phospholipid tails, and the heads. To help you with receptor protein, remember that a receptor protein passes all the way through the membrane, but it does not have a channel in it. A marker protein is found only on the surface of the membrane and does not pass through the membrane. And finally, transport proteins also pass all the way through the membrane, and they often have passageways like channels that pass through them. I'd like Mrs. Termenstein to turn off the DVD for just a moment and give the students some time to label this drawing. When she sees that you're done, she'll start the DVD again. This is a drawing that you saw the other day in class. You're looking at a photograph of the lipid bilayer. It's labeled plasma membrane. And then below that line you see the inside of the cell. Within the cytoplasm, underneath that plasma membrane, you can see a little circular structure called a vesicle. This vesicle is also surrounded by a lipid bilayer. There's a word that I've mentioned on page 2 of your worksheet called lumen. Any inside hollow area, like the inside of that circle, is called a lumen. And then I've given you a couple of other examples of lumen on your worksheet. I'd like you now to turn to page 2, and I'd like you to label parts of the vesicle and the area within and around the vesicle. I want you to label the lipid bilayer, the cytoplasm. Remember, the cytoplasm includes the vesicle, so you want to label the cytoplasm outside of the vesicle, and the vesicle lumen, and that's the hollow area inside the vesicle. Now, of course, that vesicle is not empty, it's not hollow. There's something stored in there, but it is a hollow area that has things stored in it. So now I'd like you to label the structure, the photograph, on page 2. Turn off the DVD and let the students work on that for a few minutes. Pages 3 and 4 of your worksheet have photographs that include the nucleus. Here you see a photograph of the nucleus. This is one that I showed you in class last week, or earlier this week. The nucleus is the great big circular structure. If you look real closely, you'll notice that it has a double membrane around it. It's rather light on the inside, but there's one very dark spot. And if you look at the smaller photograph on page 4, you'll notice that that small dark spot is called the nucleolus. We have not talked about that yet. The chromatin, which is made of DNA, is found all over the inside of the nucleus, and you can also see that labeled on the small drawing on page 4. Outside of the nucleus is the cytoplasm, and there we can see all kinds of things that we haven't talked about yet. So just wait on endoplasmic reticulum and mitochondrion. You can come back to these later, but for right now, I'd like you to label pages 3 and 4, and I've told you underneath or next to each of the photographs what I would like you to label. On these drawings, just like on page 2, there's no line pointing to the different structures. I guess there is on the first photo on page 3, but you'll have to draw your own lines to the structures that you're trying to label. The second photograph on page 3 is a little bit unusual. This photograph was taken with a scanning electron microscope, and you're looking at the nucleus from the outside in 3 dimensions. Okay, now turn off the DVD and let the students work on pages 3 and 4 for a few minutes. This is a photograph of the inside of the cytoplasm. Remember, in the cytoplasm there are lots of different organelles. The parallel lines that you see is part of the endoplasmic reticulum, or we sometimes just call it the ER. Right next to those parallel lines, you see hundreds and hundreds of tiny little black dots. Some of them are attached to the endoplasmic reticulum. Others are floating freely in the cytoplasm up above the endoplasmic reticulum. All of these dots are ribosomes. At the bottom right of the page, on page 5, you can see a drawing of what a ribosome looks like if you could magnify it a lot. Now I'd like you to label page 5, the ER, a ribosome that's attached to the ER, and a ribosome that is not attached to the ER. Take a minute now to label page 5. This is a photograph of a mitochondrion. This is magnified quite a few times. Notice that the mitochondrion is only about 7 or 8 tenths of a micrometer long. Mitochondria can often be recognized because they seem to be a darker organelle than most of the things in the cell. This particular mitochondrion is shaped a little bit like a foot. Notice the endoplasmic reticulum, the parallel lines outside of the mitochondrion. Inside the mitochondrion, you see some crossing lines that look very much like ER. These are membranes on the inside of the mitochondrion. Look at the drawings on page 6. At the bottom right, you can see a drawing of a mitochondrion. You can see that it has an outer membrane and it has an inner membrane that cuts into the middle of the mitochondrion and forms those crossing walls. I'd like you to label page 6 now. Notice that in each of the photographs there's only one mitochondrion, so when you label them, use the singular form, mitochondrion, and not the plural form, mitochondria. Also, in the left-hand photograph, I'd like you to label the endoplasmic reticulum. take a minute now to label page 6. There's a structure inside the cytoplasm called the centrosome, and the centrosome is made of these two structures, which are called centrioles. The centrioles lie at right angles to one another, and they're made of a lot of little tiny microtubules that run the length of each centriole. The drawing of the centriole is easier to recognize than the photograph. I'd like you to look at page 7, and you can see the drawing of two centrioles at the top of page 7, and then there's a photograph showing the centrioles in the cytoplasm of the cell. The very dark circle is a centriole that is lying perpendicular to you, so you're looking into the middle of the centriole, and the centriole on the left you're seeing from the side. The little microtubule at the bottom of the photograph is something that the centriole makes. Now I'd like you to label what you can on page 7. This is a photograph of a Golgi apparatus. The one on the left is the easiest one to recognize. You see a number of black parallel lines that are curved. These lines have little bubbles at the end. These lines are membranes that are packed together. And the Golgi apparatus will always have this kind of an appearance. Now I'd like you to look at page 8, and you can see a drawing of a Golgi apparatus on the right. I'd like you to label the Golgi apparatus in the photograph on the left, and I'd like you to label one vesicle. Remember, the vesicles are the membrane sacs that look like little bubbles. Label page 8 now. This is a drawing of a single-celled organism called a paramecium. Surrounding the body of the paramecium are a lot of little hair-like structures called cilia. In the singular, one of these hairs is called cilium, but in the plural, the cilia altogether are called cilia. Don't pay attention to the black arrows at the top and at the right, but the arrow at the left is pointing to some of the cilia. Look at the drawing on page 9. You can see the same organism, a paramecium, in the top left corner of the page. I'd like you to label the cilia on the other two photographs. I'd also like you to label the cell membrane. The cell membrane is the thing that the cilia are attached to. Label page 9 now. These cells have very long hair-like structures called flagella. A, the cell on the top, has a single flagellum, while B, C, and D all have multiple flagella. I'd like you to turn to page 10 and label the flagellum on one cell when there's only one of them, and label the flagella on the other cell when there are more than one. Label page 10 now. That's it for the part of the worksheet that I'm going to walk you through today. I'm not going to assign anything else, but if you think that you're capable of doing some labeling on pages 11 and 12, you can try that over the weekend. These are both animal cells, and just like the animal cell you see in this photograph, you can probably recognize things like the nucleus, endoplasmic reticulum, mitochondria. Certainly you can recognize the plasma membrane, and maybe some other structures as well. You could also label the cytoplasm and things inside the nucleus. So it's kind of an optional assignment to work on 11 and 12. If you haven't done so, you can also go back to page 4, and you can label the endoplasmic reticulum and the mitochondria there. I think this is about where we're going to stop with this worksheet. We're not going to do anything with page 13 right now. This is a plant cell, and we haven't talked about plant cells. So you can take a few minutes finishing up and put this away. Make sure you bring it back to class on Tuesday.